Assalamu alaikum warahmatullahi ta'ala wabarakatuh. This is the seventh and last lecture in lipid metabolism. It is not about metabolic pathways reactions, but particularly about lipid transport. We need to know how lipid is transported between different tissues. The objectives of this lecture is to discuss the rationale for lipid transport, list the chemical classes of plasma lipids, list the members of the lipoproteins and identify their main differences, describe the traffic of lipoproteins, describe typical structure of lipoprotein, describe the location, function, cofactors, and regulation of lipoprotein lipase, and finally, to identify the relationship between HDL and atherosclerosis. Dietary lipids and endogenous lipids, lipids that are synthesized de novo, has to be transported between tissues and organs for utilization and storage. So, for example, a lipid has been produced in the site of production, whether it is in the liver or in the intestine, for example, it has to be transported for another site or tissue for utilization or for storing it in, for example, in adipose tissue. So transport is important. The problem is that a lipid is insoluble in water, and the blood is mainly an aqueous environment. So how the body manages to transport lipid, the insoluble lipid, in the soluble circulation, where the environment is mainly aqueous, is by in the form of lipoproteins. And the mainly non-polar lipids are the triacylglycerol and cholesterol esters, while amphipathic lipids are phospholipids and cholesterol. The transport is much easier. Plasma lipids exist in the following chemical classes, mainly triacylglycerols, phospholipids, cholesterol, cholesterol esters, and less than 5% of the total lipids in plasma are in the form of free fatty acids. They account for the least category, but they are the most metabolically active class. What are lipoproteins? Lipoproteins are, each lipoprotein is considered a biochemical assembly of both proteins and lipids that functions in transporting lipids in blood or plasma. There are variable classes and amounts of lipids in each lipoprotein, and lipoproteins have both physiologic and clinical importance. Lipoproteins differ mainly in their density and in their electrophoretic properties. Regarding the density, lipoproteins can be separated by ultracentrifugation. And as you know, pure fat is less dense than water. So the more the lipid proportion or content of a lipoprotein, the less the density it has, and vice versa. Electrophoretic properties, lipoproteins can be separated by various types of electrophoresis. We have four major groups of lipoproteins: chylomicrons, very low density lipoprotein, VLDL, or also known as pre-beta lipoprotein; low density lipoproteins, LDL or beta lipoprotein; high density lipoproteins, HDL or alpha lipoprotein. And this is lipoprotein separated by agarodial electrophoresis. This side here represents the density, and this side here includes the names of the different lipoproteins. And as you know here, closer to the origin, there is separation of chylomicrons, which has a density of less than 0.6, compared to high density lipoproteins, HDL, high density lipoprotein, with a density that ranges from 1 to 1.2, travels farther down the separation spectrum. Why chylomicrons are here and HDL is here? Because chylomicrons, both of them, all of the four lipoproteins, contain different or varying proportions of both lipids and proteins. But chylomicrons, because they contain more lipids, they are the least dense, while HDL have high density and they comparatively have low proportion of protein to lipids. The typical structure of a lipoprotein, it contains two components, lipid and protein. The lipid component, the core, is mainly contains non-polar lipids, mainly triglycerols and cholesterol esters, while the surface lipids will be mainly the amphiphasic lipids, as we said, the phospholipids and the cholesterol, oriented with their polar parts near the aqueous medium. Protein moiety, also called apolipoprotein part of a lipoprotein, less than one or less, at least one apoprotein will be present in each lipoprotein. It could be more than one. The proteins can either integrate, like apolipoprotein, or it could be preferred, that is transferable between different lipoproteins like apoprotein C. Protein components are characteristics of their lipoproteins, and there is variable amounts of protein content. For example, as we said, HDL, which has the highest density, has 60% of its content as protein compared to only 1% protein content in chylomicron. And this is a cross section of a typical lipoprotein. It is almost globular, and as you can see, this is the surface that is facing outside the aqueous environment, and this is the core of the lipoprotein. The core contains the non-polar lipids, mainly cholesterol esters and triglycerols. Lipid content of the surface are free cholesterol and phospholipid, and as you can see, their heads, the polar part of their molecules is facing outside, while the non-polar tail is facing inside. The blue parts here represent as two forms of proteins in lipoprotein. Either integral part of the structure, it is fixed, it's not transferable, such as apoprotein B, and as well as the integral, the peripheral types of apoproteins, such as ApoC, which are present on the surface and can be transferred between two different lipoproteins. And the nomenclature of apolipoproteins follows an ABC pattern. ApoA, we have four types of ApoA. It is present mainly in HDL and chylomicron. ApoB, we have two types, ApoB100 and ApoB48, is present in LDL, VLDL, and chylomicrons. It is a glycoprotein because it contains monosaccharides such as mannose, galactose, glucose, and glucose. ApoC, present in three forms, C1, C2, and C3, is freely transferable between several different lipoproteins, for example, VLDL and HDL. ApoD is a subfraction of HDL. ApoE is present in various lipoproteins. It is contains a higher content of arginine amino acid, yes, about 10% of its content is arginine. The functions of lipoproteins, mainly they form part of the structural lipoprotein, so they have a structural role, but they also act as enzyme cofactors. For example, ApoC2 is a cofactor for the enzyme lipoprotein lipase. ApoA1 is a cofactor for lysine cholesterol acyltransferase. And apolipoproteins also act as ligands for interaction with lipoprotein receptors. For example, apolipoprotein B100 and ApoE are ligands for LDL receptor, while ApoE is a receptor for renin, chylomicrons, for example, and A1 is a ligand for HDL receptor. What about albumin and free fatty acid? When we say free fatty acid in plasma or in blood, they are not actually free. They are transported within albumin. So albumin helps in transporting free fatty acids in circulation. And the free unsaturated free fatty acid, of course, arise from lipolysis of adipose tissue, from breakdown of triacylglycerol stored in the adipose tissue. And the rate of lipolysis determines plasma concentration, which in turn determines uptake of free fatty acids by tissues. Free fatty acids are not actually free, as we said, and they are low in the steady state, logically, and high in the fasting states, as well as in diabetes mellitus. Membrane free fatty acid transport protein is a protein that transfer free fatty acids across the plasma membrane. And then inside the cell, which is also aqueous, there is another binding protein that helps in binding free fatty acid within the cell. We will discuss chylomicron and VLDL together because they have so many similarities. Both chylomicron and VLDL transport triacylglycerols. Chylomicron will transport triacylglycerol from the intestine, where it is absorbed from the meal, and it will transport it across the circulation to different tissues. VLDL will transport triacylglycerol from the liver to extrahepatic tissues. There are many similarities in the formation of chylomicron and VLDL by the intestinal cell, which of course synthesizes chylomicron, and parenchymal hepatic cells, which synthesize VLDL lipoproteins. The similarities include their synthetic pathway. It starts by synthesizing the lipoprotein. For example, here is an intestinal cell and here is a hepatic cell. The synthesis starts in the rough endoplasmic reticulum of ApoB 100 in the or ApoB 40. It depends on whether it is in the intestine or in the liver. And then following the synthesis of ApoB, it will be the lipid part to form a whole molecule of lipoprotein will be added in the rough endoplasmic reticulum, as well as any sugars, and then it will proceed to Golgi apparatus to adhere the Golgi apparatus to add the carbohydrate if needed, and to package it into secretory vacuoles, which will attach to the membranes and release their content. Chylomicrons from the intestine will be released in between the intestinal cells here, and then it finds its way into the lymphatic, where it will be carried through the lymphatic system to join the general circulation, and then from there it will head mainly to the liver. The joining of the lymphatics with the general circulation, of course, is through the thoracic duct. In the hepatic cell, the same pathway of synthesis from the rough endoplasmic reticulum to the smooth endoplasmic reticulum to Golgi apparatus to the secretory vacuoles. VLDL here will be released in a space called space of VII in between the hepatic cells and the hepatic sinusoids, and then from there it will join the circulation through the lumen of blood sinusoids. So chylomicrons will be carried first through lymphatics and then to the circulation, while VLDL from the space of VII through the sinusoids, it can join the blood directly. Chylomicrons and VLDL are widely categorized in a minimum of minutes to up to less than 1 hour. The majority, up to 8%, will be categorized by the adipose tissue, heart, and muscle. So these are the tissues that are target tissues for chylomicrons and VLDL. The liver does not catabolize chylomicrons and VLDL significantly. Why? Because because as the enzyme, the lipoprotein lipase is needed and is not active in the liver. Lipoprotein lipase is needed for hydrolysis of chylomicrons and VLDL at the triacylglycerol content of both chylomicrons and VLDL. Hepatic lipase, in fact, does not readily react with chylomicrons, particularly However, it can deal with chylomicron remnants and HBI. Lipoprotein lipase, where it is located, it is anchored to the capillary endothelium by a heparan sulfate. We know heparan sulfate, a glycosaminoglycan, negatively charged proteoglycan. As the lipoprotein lipase, it is not free in the blood in significant amount. It is mainly linked to endothelium, and it is released when it is anchored by injectable heparin, which is clearing of IPMia. So it is an iatrogenic process. Normally, lipoprotein lipase is linked to the endothelium. It needs cofactors, particularly phospholipids and ApoC2. So the gyromicron and VLDL will provide the substrate for the lipoprotein lipase, as well as a cofactor for its action. What is the action of lipoprotein lipase? Hydrolysis of triacylglycerol that is contained within chylomicrons and VLDL. We said tissues that are concerned with triacylglycerol hydrolysis in chylomicrons and VLDL are the heart, adipose tissue, and muscle. However, heart and adipose tissue have different Km values. The Km is low in the heart and it is 10 times greater in adipose tissue. So the heart and adipose tissue will have different affinities for triacylglycerol. So in the transition state from the feed to the start state, where the plasma concentration of triacylglycerol will be low, cardiac lipoprotein lipase will still be saturated and functioning and providing lipids for the heart, while at the same time, because of the low concentration of triacylglycerol as a substrate and the high Km of adipose tissue lipoprotein lipase will not be saturated. It will be reduced. The action will be reduced, and this will help in directing all or most of the available triacylglycerol for to be used as a fuel source by the heart. So when there is low free fatty acids or triacylglycerols and low energy and no the body is in the fasting state, which body needs more energy and more sustainability? The heart, of course, because adipose tissue is mainly for storage. Insulin and hormonal regulation of lipoproteins is mainly through insulin and glucagon. Insulin enhances synthesis of lipoprotein lipase by adipocytes and its translocation to the luminal surface of the endothelium. Chylomicrons and VLDL are hydrolyzed while still attached to the enzyme on the endothelium. So they act while still attached. There is progressive hydrolysis of triacylglycerols to diacylglycerol to monoacylglycerol to free fatty acid and glycerol. The bulk of free fatty acid is taken inside the tissue. Some free fatty acids are released into the circulation. Glycerol as well is released into the circulation. And as a result of hydrolysis of triacylglycerol of chylomicron and VLDL, what we are ending up with, chylomicron will be converted to what we call a chylomicron remnant, while VLDL will be converted to IDL, intermediate density lipoprotein, which is a remnant of VLDL. Both remnants of chylomicrons and VLDL are smaller than their original or parent lipoprotein, and they are deficient in triacylglycerols because 90% is hydrolyzed by lipoprotein lipase, and they are relatively enriched in cholesterol and cholesterol esters because comparatively the concentration of the lipid content, particularly cholesterol and cholesterol ester, will be higher. They are the same amount, but comparatively, because the triacylglycerol is lost, their percentage the percentage of cholesterol and cholesterol is higher. And because of hydrolysis, ApoC will be lost, but ApoE will be still present. This make the remnants of both chylomicron and LDL will be taken up by the liver via the receptors LDL and LDL-related receptor LPR. And this is to show you here the fate of chylomicron. Chylomicron, as we said, is formed by the intestinal cells. It finds its way into the lymphatics and then via the thoracic duct to the circulation. Initially, it is called a nascent chylomicron that contains triacylglycerol and cholesterol in the center. It contains ApoB48 and ApoA. And then it can obtain this nascent chylomicron obtains ApoC and ApoE from HDL. So it is now it contains 4 types of ApoP, apolipoproteins, B48A as well as C and E. Now it is called a mature chylomicron as compared to the nascent chylomicron. And this goes into the extrahepatic tissues while a lipoprotein lipase hydrolyzes the triacylglycerol content. The free fatty acids will enter circulation, will enter the extrahepatic tissues into the inside of the cell. Glycerol will be released. What is left from the chylomicron is a chylomicron remnant. ApoA and C will be returned to the HDL. This chylomicron remnant is taken up by the liver by 2 types of receptor, as we said, the LDL receptor, which uses ApoP100 and E as cofactors, and lipoprotein related protein receptor LRB to release fatty acid and cholesterol in the liver. And this is to show the metabolic fate of VLDL, which is quite similar to chylomicron. However, the initial site of synthesis of VLDL is the liver. We have nascent or newly formed VLDL. It contains P100, cofactor, and apolipoprotein E and C, and the core of the VLDL contains triglycerides and cholesterol. It obtains ApoC and ApoE from HDL. Now we have the mature VLDL, which carries triacylglycerol to the extrahepatic tissues, where lipoprotein lipase releases triacylglycerol or hydrolyzes triacylglycerol, freeing free fatty acids to be taken up by the extrahepatic tissues with the release of glycerol. ApoC is obtained by HDL. We have left with the LDL remnant, which forms LDL, and LDL is either taken up by extrahepatic tissues through an LDL receptor or taken up by the liver through also an LDL receptor. What is LDL? LDL is a cholesterol-rich lipoprotein that results from metabolism of VLDL. So VLDL will initially form IDL, intermediate density lipoprotein, which in its turn form low density lipoprotein LDL. Most of the cholesterol of VLDL is retained within IDL, which is taken up by the liver, or converted to LDL, which is taken up by the liver, 30%. However, the majority of LDL, 70%, will be taken by extrahepatic tissues. The significance of LDL is that there is positive correlation between atherosclerosis, the incidence of atherosclerosis, and the concentration of plasma LDL. The higher the plasma LDL, the higher the risk of atherosclerosis formation and the risk of ischemic heart disease. There are 2 types of receptors for LDL. LDL receptor, which uses ApoB100 and E as cofactors, is highly regulated and is specific for B100 cofactor, but not for the B48 that comes from the intestine. And another receptor for LDL is a scavenger receptor. However, this receptor is not subject for regulation. LDL receptor is a glycoprotein that spans the membrane and is located in bits within the membrane. LDL is taken up intact by endocytosis, and then it will be broken down in lysosomes where hydrolysis of cholesterol esters and apolipoproteins takes place. The number of LDL receptors is regulated by cholesterol requirement, how much the body needs cholesterol. HDL lipoprotein is also a cholesterol-rich lipoprotein. However, its relationship with ischemic heart disease and atherosclerosis is the opposite of that of LDL. HDL is synthesized in both liver and intestine. The nascent HDL, the newly formed HDL, consists of a discoid phospholipid bilayer. It contains ApoA, but it does not contain C or E. It contains free cholesterol. Liver is the final destination for destruction of HDL cholesterol esters. What are the functions of HDL? HDL is a sort of a storage or a repository for ApoC and E, which are needed for the metabolism of chylomicron and VLDL. We said the nascent chylomicron and the nascent VLDL does not contain C or E apolipoproteins. They obtain them from HDL. So HDL here function as a store or a source for ApoC and E. HDL also functions in what we call reverse cholesterol transport, and that is a transport of cholesterol from tissues to the liver, uptake and esterification of cholesterol by HDL3, which becomes bigger and less dense, forming HDL2, and then hepatic lipase hydrolyzes HDL phospholipids and triacylglycerols, which allows release of cholesterol esters, forming the less dense HDL3. Apolipoprotein A1 is released into the circulation. It can associate with minimal amount of phospholipids to form a pre-beta HDL. Pre-beta HDL is the most potent form of HDL in inducing cholesterol efflux from tissues to form discoid HDL, which then takes up more cholesterol to form HDL3. This will be more clear as we come to the figure in a couple of minutes. HDL2 concentration is inversely related to the incidence of coronary atherosclerosis. So this reflects the efficiency of cholesterostatic scavenging from tissues because it functions in reverse cholesterol transport. It scavenges or it removes or it clears cholesterol from the peripheral tissues, from the extrahepatic tissues back to the liver. What about HDL1? HDL1 is found in diet-induced hypercholesterolemic animals, so it is in laboratory animals. It is rich in cholesterol, and it has one type of lipoprotein, and that is lipoprotein E. HDL1 can be taken up by ApoE remnant receptors and LDL receptor. So this is a discoid form, the HDL, the nascent HDL synthesized in the liver. HDL, as you can see, it does not contain ApoC or ApoE. Okay. The main lipids here are phospholipids and cholesterol. It can also be synthesized in the intestine. So this is discoidal HDL. HDL can take cholesterol from the tissues, so now it contains cholesterol in addition to cholesterol esters and phospholipids to form HDL3. HDL3 can take up more cholesterol to form HDL2, which as you can see is bigger, however less dense than HDL3. HDL2 now heads up to the liver, where it is subjected to hepatic lipase to release cholesterol esters, and phospholipids. And this is what we mean by reverse cholesterol transport from the extrahepatic tissues back to the liver. What is the biomedical relevance of lipoproteins? We have what we call lipoproteinemias, which can be either primary or secondary. These lipoproteinemias include defects in lipoprotein formation, transport, or destruction. However, not all these lipoproteinemias are harmful. Some forms are more harmful or even beneficial. The most important form of disorder that is related to lipoproteins is, of course, familial hypercholesteremia. Hypercholesteremia, so the level of cholesterol will be high in the blood due to defective LDL receptor. A rare form of disorder is A-beta lipoproteinemia. A-beta lipoproteinemia, as the name implies, A refers to absence or deficiency. So beta lipoprotein will be deficient or absent. It is due to defective loading of ApoB within the lipids. No formation of chylomicrons or VLDL, because both of them contains ApoB as an important part of the structure of both chylomicron and VLDL, and this will lead to accumulation of lipids in both the liver and intestine. This concludes our talk. Thank you very much.